Thank you very much. Hello, everyone. I'm very honored to be here today, and I thank very much the organizers for this invitation. Before going to our preferred system that happens to be a eukaryote instead of a bacterium, I would like to go as well back to some reading of François Jacob and try to present you what is the general question and the way we approach the evolution of social behavior in microbes. So in general, as Jacob said, in biology, we face different levels of organization, and we want to connect the properties that exist at the level of single units that might be cells to the variation over evolutionary time of the frequencies of these units that possess different qualities and how they change. And this change is affected, of course, by evolution. So in doing this, there's a two-way dialogue. First of all, one has to understand the logic of the organization of single units into populations or higher structures. And on the other hand, we want to understand what are the evolutionary feedbacks that adaptation imposes on the single units properties. So for, let's say, paradigmatic multicellular organisms, this goes through a step that is basically development and how do a zygote turns into a grown-up organism. And this connection is sort of facilitated by the fact that it is the program, always quoting Jacob, that is coded in the genes that creates, that determines the developmental program, and therefore the function that the organism has. This is a bit more problematic or difficult in microbial multicellular collectives because because microbes tend to occur embedded into multicellular or pluricellular structures that vary a lot, both in size and in composition. And therefore, the properties or the functions of at this level, they are not all that easily deduced from the properties of the underlying cells, from the cells that constitute these collectives. The existence of this heterogeneity within microbial collectives or within, if you want, social manifestations of microbial life makes so that there are possibly conflicts that emerge due to the fact that different kind of cells, they can reproduce independently within a collective. And for this reason, people think of the evolution of society in microbes as being conceptually framed by sociobiology. So the reasoning behind this is that these cellular collectives have multiple functions that in general require cells to invest into this function, be it the production of a matrix or the production of, I don't know, coalescence for bacteria. These functions, therefore, they are threatened by the existence of varieties, variants that might not contribute to this collective function, and these are commonly called cheaters. And these cheaters, they have a fitness advantage with respect to the cells that contribute. As a consequence, one might predict that the function might be disrupted in the long term on the evolutionary timescale due to the spread of those cheaters. And then the question becomes, how is the collective function maintained over evolutionary time? Okay, let me be more concrete now, and I will introduce the life cycle of Dictyostelium discoideum, that is also known as the social amoeba. This amoeba lives most of its time as a unicellular amoeba in the soil. And so cells don't display any multicellular behavior until they run out of food. But when they are starved, they start entering into this multicellular facultative life cycle. Cells aggregate, and this multicellular aggregate undergoes a series of steps of development, of multicellular development, until it turns into a fruiting body where part of the cells die, forming a stalk. And this stalk is necessary to lift up a ball of spores that get dispersed and then seed the next generation. So you see that in order for efficient dispersal, one needs some cells to sort of sacrifice themselves. And this is why this system is typically viewed as a case of altruistic division of labor. You may think that this is basically no different from the germline soma distinction in paradigmatic multicellular organisms, but these microbes have another peculiarity, that is that they share with a number of other unicellular organisms that evolved the same kind of life cycle independently multiple times in the tree of life. So as you can see in this movie, so the feature of this aggregative life cycles is that the multicellular aggregate originates from the coming together of different cells. So it's not grown clonally from a single cell. And so many cells of different origin, they can enter the same aggregate and then undergo development. Also, if here you see the slugs, for instance, forming, also if the aggregates are chimeric. So chimerism is very common in this kind of organisms. Therefore, this calls for an explanation, as I said before, of why cheaters do not take over everywhere. Now, if we want to be a bit more quantitative on this kind of question, then we can do some experiments in the lab. So chimeras are very easily formed in the lab, and they are found also in the wild. And we can mix two different kinds of cells at a given proportion that we can control. These cells then co-aggregate, and then we can measure what is the proportion of cells of the two types in the spores, and then compare these two proportions. In this way, we can detect whether there are certain populations that get overrepresented in the spores with respect to what would be expected. This is quantified by what is called the spore bias. And I want to stress that spore bias in general is of one strain or one population relative to another one. So it's not an absolute value. And in general, strains that have a positive spore bias are called cheaters. And these cheaters, let me explain more in detail why this is because if you think of measuring the spore bias as a function of the proportion, the initial proportion of here the red strain, and then you start with mixing a small amount of red cells, then if the spore bias is positive, spores will have more red cells than would be expected by the initial composition, and therefore the next generation will have more red cells. And if you keep going like this, you get to an evolutionary solution whereby you expect to find only red cells. And this solution is known in evolutionary game theory and also in sociology as the tragedy of the commons, that tells that any time that there are this kind of conflicts, then the null expectation is that cooperators should basically be eliminated. In practice, in Dictyostelium, it is known that there are some extreme cheaters that are unable to form a multicellular fruiting body on their own, and they need indeed the presence of cooperators in order to go through the multicellular cycle. Now, so sociobiology found a number of ways by which this kind of evolutionary trajectory can be avoided. My focus today will be a bit different, and what we want to understand is what underpins this spore bias profile. And there are different hypotheses in the literature, even though it's not really phrased in this way. So the first hypothesis that was basically the initial hypothesis of people who started using Dictyostelium discoideum to study the social evolution in microbial communities was that the spore bias was genetically encoded. And therefore, strains or species could be sort of classified based on their meanness or their being cooperators. And one could even establish a hierarchy that went from strains that were better cheaters to those that were better cooperators. This view was already shown to be not so accurate because these hierarchies seem to depend a bit on the context, but then this context dependent was clearly shown by observing that spore bias, or if you want stock investment that is equivalent, depend on the frequency of strains in a population and therefore on the social context they encounter. And in this respect, from a theoretical viewpoint, the authors proposed that what is coded in the genome is not really the fact of the amount of cooperation that we invest, that a strain invests, but rather the capacity of responding to a given context. At the same time, other studies that were more on developmental biology show that the final fate of a cell is influenced by the phase of the cell cycle that this cell has at the beginning of the aggregation. And therefore, it might depend, actually, the social behavior might depend on factors that are completely out of control of a cell, because this would be like a lottery model. The fact that you become a spore is sort of independent of your genes. And I want to stress again, going back to the observation by Jacob, that what actually reconciles ecology and evolution, or if you want, development and evolution, is heredity. I want to stress that in going from here to here, the amount of inheritance of the social behavior decreases, and as a consequence, the predictive power of a measure like spore bias decreases. So one needs to understand where do real systems are located along this gradient in order to understand to what extent social behavior can be selected. So this was the subject of the thesis of Mathieu Forget, who is now at ETH. And he proceeded in 3 steps that I will drive you through. First of all, he wanted to find a way to control phenotypic bias in chimeras, both isogenic and heterogenic. And then he wondered whether it was possible to find some individual cell basis of these biases. And finally, if I have time, I will tell you something about some models for group formation in heterogeneous populations. So it is well known since quite a while that phenotypic differences can induce spore biases. But these phenotypic differences are usually induced by pretty aggressive means, like adding drugs. And what Mathieu did was to have a more, let's say, a smoother way to change the phenotype, and in a way that the phenotype could be controlled quantitatively and continuously. So he decided to take populations of Dictyosteliums at different stages of their demographic cycle. So in early exponential phase, mid and late exponential phase, and in early stationary phase. And our ansatz was that these different phases corresponded to some phenotypic difference. And then we could make what we call chronochimeras, that are mixtures of pairs of populations that are harvested at different demographic stages. So then one can quantify by flow cytometry the composition, the initial composition of the population, and the composition of the spores after a development of 24 hours. Oops, sorry. What Mathieu found was that depending on the timing of the initiation of aggregation, spore bias profiles change not only quantitatively, but also they can change qualitatively. That means that, first of all, you have these biases, so these cells are isogenic, so we would nonetheless classify some populations as cooperators and some others as cheaters, and this classification is not consistent when one changes the different timings. So the amount of bias that he observed is comparable to the kind of bias that one measures when mixing different strains. So the next question was, does this variation adds to the variation that is induced by genetic differences, or it just disappears when one considers genetic chimeras? So the answer is that it depends, but at least in cases where cheating is not very extreme, Mathieu found that when two different strains, these are two different lab strains, were mixed at a synchronous timing, then he could observe like a frequency-dependent kind of bias, whereas when the two strains were mixed so that one was harvested early and one late, or vice versa, then the behavior, the social behavior, would have been classified as cooperative or as cheater, depending on the timing. So let's say the genetic determination of the social strategy, therefore, seems to be variable depending on the context, and varying even when such subtle changes as those induced just by growth are considered. Now, what we wanted to do was to understand why could differences that were present at a very early stage in the multicellular cycle propagate throughout all the development until the spores. And so to us, it didn't make a lot of sense because Dictyostelium changes a lot its gene expression during the multicellular cycle. So we thought, well, perhaps this has to do with cells who do not enter groups. So these cells that have been termed loners and have been argued to be extremely important to explain evolution in unpredictable environments can be simply scored under the microscope. And this is what Mathieu did, and he found that populations that had a higher propensity to leave cells outside were also those that had produced less spores. This is quite logical, but then what makes so that a given population at a given growth stage leaves more cells outside? And in order to understand this, he looked at single-cell trajectories in populations at different stages of growth. And he found out that, in general, populations, isogenic populations, are composed by two subpopulations of cells that are motile or they are hardly motile. And these proportions change during the growth, demographic growth, whereas the properties of these two populations do not seem to change a lot. So somehow populations that tend to leave more cells outside seem to be those that have a higher proportion of cells that are less motile. And now we are trying to understand more about these differences in motility. Where do they come from? Are they related to a phenotypic switch, or are they something that has longer-term memory, like epiphenotypes? We still don't have an answer, but Alexandre Souchard, who might be here in the audience is now doing very accurate work to try and classify these different kinds of motility by using tools from physics and tracking of particles. Okay, I want to finish just with 2 words about some mathematical models or numerical models that we have developed to try and reproduce these experimental observations. So first of all, we used models inspired by active matter physics where cells are modeled by particles that can be chosen to have different motility. And then we studied whether these differences in motility were enough to obtain this kind of frequency-dependent biases that we observe in experiments. And this is the case, and there are some very interesting regimes where actually the evolutionary prediction would be a coexistence of cells of different motility. And finally, in a more coarse-grained model, that is more of the evolutionary game theory type, we explored what would be the use of having different motility in an evolutionary time, on an evolutionary time scale. And we found that if cells are supposed to use their motility in a different way when they are in a social context or when they are alone, then one could predict the evolutionary emergence of life cycles that look very much like aggregative life cycles. And these life cycles allow to compartmentalize conflicts so that the social phase where conflicts play out is temporally separated by the phase where cells live on their own. And with this, I will conclude by reminding that, so we showed that contingent single-cell phenotypic heterogeneity modulates social strategies. And so the attribution of social strategies to genotypes might be more tricky than it has been considered so far. And that the multiplicity of these sources of single-cell variation may keep populations far away from the kind of evolutionary optimum that would be predicted just based on the idea that social behavior is purely based on genes. And perhaps this may also pave the way to the coexistence of different types and understanding why division of labor is so common also in microbial populations. And with this, I would like to thank my collaborators, so Mathieu, Sandrine, and Alexandre, mostly on the experiments, and the two Leonardos for the theory. And thank you very much for your attention.